I remember when I was a veterinary surgical student, our esteemed professors and many of our classmates believed that it was impossible to learn surgery without harming animals. Surgery could be learnt in no other way than by practicing surgical procedures on healthy animals which were then killed at the end of the procedure. Unfortunately, this is the traditional way that students learn surgery in veterinary courses around the world, across Australia, across North America, in many countries, oddly and to its credit, not the United Kingdom, where for decades students have learned surgery by assisting with beneficial surgeries on real patients out in private veterinary clinics and even animal shelters. But in most veterinary schools, certainly in Australia where I trained, this was the standard way for learning surgery. But I was sure there must be humane alternatives to this kind of training, because I went into veterinary school to learn to become a healer of animals, obviously, rather than a killer of animals. So what I did was I went out into private veterinary clinics and animal shelters and was involved in spays and neuters, sterilisation procedures on animals that were awaiting adoption in the shelters, to ensure that when they were adopted they wouldn't go on to continue breeding and contribute to the pet overpopulation problem the shelters were trying to solve. So because of going out into these animal shelters and the private veterinary clinics, I ended up with around about 5 times the amount of surgical experience of my classmates who killed to obtain their degrees. And our program was an outstanding success. The animal shelter sterilisation was mainstreamed into the general curriculum soon after that because it was such a big success. Similarly, scientists who are involved in conducting invasive research on animals, on pharmaceutical companies, often make claims that in no other way than by conducting harmful animal experiments is it possible to develop cures for major human diseases such as cancer, heart disease, stroke and so on. But if you actually look at the evidence paints a very different picture. What I've been doing for the last decade or so is conducting studies that have actually assessed how often these invasive animal experiments that are published in the scientific literature, in scientific reports and studies, how often they're actually used by scientists who are developing cures for human diseases, how often the results of those animal experiments translate into new cures for serious human diseases. And what I've done is I've looked at systematic reviews. Systematic reviews are different from normal reviews of the evidence because they aim to be very thorough. They aim to incorporate all relevant published studies, such as animal experiments in this case, and they aim to look at them all systematically, and they use multiple bibliographic databases to make sure that no important pieces of evidence are missed. They bring them all together and they try and work out the overall outcome. There have been, by myself and by other scientists, there have been 20 systematic reviews at least that have examined the question of how useful are invasive animal experiments in contributing to specific fields of human healthcare. And another 7 have looked at how predictive are they for predicting human toxicity. And out of those 27 studies, in only 2 cases did the authors of the studies conclude that the animal experiments were either significantly useful in contributing to cures for human diseases that were effective in human patients, or else are substantially consistent with outcomes in human volunteers in clinical trials. In most cases, the animal models proved to be insufficiently predictive for human beings and did not contribute substantially or were not consistent with human outcomes. And there's a whole range of reasons why the animals are poor models for human beings, which are described in my book. With respect to toxicity, none of the 7 studies looking at toxicity of important human toxicities such as the propensities to cause cancer and birth defects, none of these 7 studies actually demonstrated that the animal models were reliably predictive for human toxicity. So that's the evidence. So what are we to do if we don't experiment on animals? Well, there are a range of measures that can be used to try to develop effective clinical interventions for human patients, starting with, I suppose most obviously, simply making sure that all the evidence has been properly looked at. There's a large amount of data on chemicals, environmental contaminants, on drugs which is locked up in commercial company files and not released into the public domain because of reasons of commercial confidentiality. This has substantial implications for public health, I think, when the toxicity information is not released about various chemicals, and when treatments for human patients are potentially delayed. So I think that there need to be regulatory mechanisms to ensure more data sharing, with perhaps compensation of companies that engage in this process, and indeed this is currently being used by the European authorities within one of the large-scale chemicals testing systems within Europe. So it's certainly possible to compel companies to share this kind of information. So that's the first thing that needs to be done. After that, we can do a thorough physicochemical evaluation of particular compounds with computer simulations and computer modelling. There are structure-activity relationships, which are computer simulations that predict toxicity or other biological activity on the basis of chemical structure. There are expert systems which try to mimic the judgment of expert toxicologists based on known rules about factors causing toxicity, combined with information about a particular compound under consideration. There are a wide variety of cell cultures, in vitro cultures, things like protozoa, bacteria, yeast, mammals, and indeed human cell cultures as well, that can be used to predict a wide range of toxic endpoints, either individually or combined as batteries to increase the spectrum of toxins which are detected. And particularly when you're using human cells, you tend to get results much more quickly, much more reliably predictive for human patients, and much more cheaply than a classic 2-year rodent study, which is commonly done and not very predictive and much more expensive. So there are those available. There are enhanced trials on human volunteers to increase the safety for the human volunteers and increase the predictivity for more diverse patient populations. There are a range of measures that are listed in my book which have been described in the scientific literature as ways to improve these human clinical trials. There are surrogate human tissues that can be used. For example, human placental tissue obtained during childbirth in hospitals has a functional receptor for nerve growth factor, so it can be used in studies, neurophysiological studies, neurotoxicological, pharmacological studies, and so on. A range of advanced imaging modalities such as functional magnetic resonance imaging, PET, MRI, CT scans, and others. There are studies of human populations, sociological studies, epidemiological studies designed to try to identify factors involved in disease causation and risky lifestyle choices for human beings. So there are a wide range of measures, and indeed my book goes over these in quite a lot of detail about what the alternatives are to conducting invasive experiments on animals. What I would love would be if people would consider buying the much cheaper paperback version of my book and donating that to university libraries, high school libraries, public libraries in their area, because animal experimentation is a common student assignment topic. And when students are required to do essays or do assignments on this topic, they will go to their libraries and they'll look for information about animal experiments, and they'll find this book. And I think by reading this book, they'll learn a lot of really useful information about this topic, and a lot of really hopefully useful general scientific and biological concepts too. I've included quite an extensive glossary of the basic scientific concepts, which will be, I think, very useful for people learning biomedical and health sciences related subjects, or biological, basic biology as well.